Today. Okay, so thanks for joining. Today we have the great pleasure of having Dr. Joe Patter as a speaker. Dr. Patter, who obviously doesn't need an introduction, as many of you already know him, but I'll try to make one anyway. Dr. Patter is a graduate of Georgetown University and received his MD from Case Western Reserve University in Cleveland, Ohio, and completed his specialty training in both hematology and oncology. He also obtained an MSc in clinical epidemiology and biostatistics from McMaster University. As most of you already know, Dr. Patter was the director of the CCTG from 1980 to 2007, where he took an active part in the development, execution, and analysis of over 100 cooperative cancer trials. He was also head of the Department of Epidemiology and Biostatistics from 1987 to 2000. He held the Edith Eisenhower Chair in Clinical Cancer Research at Queen's University from 2000 to 2007. He also served on numerous research advisory bodies and selection committees in Ontario, Canada, internationally, and particularly in the UK. On his retirement from the CCTG, Dr. Patter assumed the position of emeritus professor at Queen's and subsequently completed a 3-year term at Cancer Care Ontario as the vice president of clinical and translational research. In the fall of 2013, he returned to the CCTG as a part-time senior investigator. Dr. Patter is currently the chair of the management committee for the Canadian Cancer Trials Network. He is a member of the PDQ Adult Treatment Board of the US National Cancer Institute. He also serves on the International Registration Review Committee for the UKCRC Registered Clinical Trials Unit Program, as well as the International Advisory Committee to the Leeds Clinical Trial Research Unit. It should be of no surprise that because of the profound impact that Dr. Patter has had throughout his career, that he recently received the inaugural Lifetime Contribution Prize from the Canadian Cancer Society. So again, it's a real pleasure to have him today. And it's also an honor to be mentored by Dr. Patter on almost a weekly basis. So again, Dr. Patter, it's a real pleasure to have you, and we're all looking forward to your presentation.
Oh, thank you, Pierre-Olivier. It's a pleasure to be here and to do something related to the Queen's Cancer Research Institute, which is, I have fond memories of the days of its founding and the building that we all live in now. So I'm going to talk about what I said I was going to talk about, which is the title here. And the living means I'm going to try to trace how this idea evolved during the time I was caring about how clinical trials were done. So here's an outline of the talk. I'm not going to read all this to you, but basically we're going to go back about 40 years, or actually slightly longer than 40 years, move forward to an example of a trial that embodied our idea of how we should do clinical trials, come up to the present and then back up again and then make some recommendations. So I became, as you heard, I became the director of the what was then the NCIC Clinical Trials Group, now CCTG, in 1980. And that was the year that this book was published. There was an article by these authors many years before that, but both the article and this book were the first place where the distinction between pragmatic and explanatory trials was made. And the book really expands greatly on what was in the original article. But the example they gave is actually the same in both places. And they gave this example. This is now, this is like 50 years old now, but it's almost contemporary. So there are 3 biostatisticians, and their idea was to try to distinguish between 2 ways of conducting a clinical trial. And they gave this example. Imagine there's a drug that is thought to be capable of improving the response to radiotherapy, but has no actual effect of its own. And you wanted to find out whether we wanted to study this drug. Well, you could study it in two different ways. If you really were intent on isolating the actual effect of the drug, that would be what they called an explanatory trial. You would want to give the drug for 30 days before radiotherapy, but you'd want the radiotherapy to be given at the exact same time in the two arms of the trial. So you were only studying the sensitizing effect of the drug. That's obviously not the way radiotherapy would be given in practice. But if you were interested in really finding out whether giving this drug to people who are receiving radiotherapy benefited them and wanted that the results of your study to be used in practice, you would compare your experimental arm, the 30 days of drug followed by radiotherapy, to the way radiotherapy is given in practice, i.e. immediately upon it's being needed. And so they distinguish these two ways of doing things as explanatory and pragmatic. They said that neither is better than the other. The radiotherapy is the same in both arms, but one design allows you to what they said allows you to test a biologic hypothesis. The other design allows you to make a decision about how to treat patients. So that's the basic distinction, and we'll speak about this a number of times. So this was, as I say, this book came out in 1980. I and many other, a number of us kind of digested it, talked about it, thought about it, and came up with our understanding of what these authors meant. So explanatory, as I've said, test a biologic hypothesis and really get at the underlying mechanism of how something works. Whereas pragmatic, and this is quoting from the book, is a pragmatic study is aimed at providing practicing clinicians with a basis for decisions concerning the choice of therapy. So pragmatic trial is aimed at supporting a later decision about how to care for patients. And the book expands, and as did the original article, on the implications of adopting a pragmatic approach. You have to think almost reverse engineering it. If you're a practicing clinician and you want to make a decision about whether to use something that has just been studied in a clinical trial, what are the characteristics of that trial you would like it to be? Well, you would like it to be carried out in the population that is the one you are interested in treating, and you'd want the intervention to be able to be given, and you'd want the intervention to have been compared to an existing standard of treatment. If you are going to decide whether to implement this innovative therapy, you would like to be sure that it's better than what you're currently doing. And then finally, the study, not finally, actually, there's many more characteristics, but key is that the study needs to address an endpoint that would matter to you and to your patients. So that's kind of the basic core idea of how we understood what a pragmatic trial was about. We got to put some of our ideas into writing fairly early on in the history of the group. 1986 isn't that long from 1980. You'll recognize some of the names here, but this was us back then. I don't know how many, I couldn't count how many faculty we have now, but back then we had 4 of us. And we wrote this art. This is really a letter to the editor in a journal, Cancer Treatment Reports, which actually isn't easily available anymore. The journal doesn't exist, but even you can't even find this article online. But in any case, we wrote a letter to the editor of the journal because previously the then head of the cancer therapy evaluation program in the U.S. had written an editorial criticizing the cooperative groups, of which we were a cancer cooperative group, but not at the time part of the US NCI activity, criticizing them for not dealing directly with some key pressing questions about treatment and specific the hypotheses about what might work in treatment. And interesting enough, a couple of those ideas, the somatic mutation therapy is what we knew as the Goldie-Coleman hypothesis, and high dose drug administration was dose intensity, both Canadian ideas. So as it turned out, we had done some studies that at least those ideas contributed to how the studies were designed, and found in those cases and in some other cases that there was a bit of a tension between testing a biologic hypothesis and making sure you answered a clinically relevant question. So in this letter, we cited the book that I just showed you, and mentioned its distinction between explanatory and pragmatic trials, and pointed out that there had been some articles quoting this book, but most of the articles, most of the people who wrote about what these authors talked about, focused on their argument, which is, I think, has been clearly accepted, is that for a trial that is pragmatic in design, the final analysis should be what's called intent to treat. That is, all patients who are randomized to each of the arms are counted as if they'd received the treatment on that arm, whether or not they actually did. And the concept behind that is that when you take the results of a trial and implement them in practice, you don't know whether the patients will cooperate. So it's really what's being tested in the trial is the intent to give a treatment rather than the treatment itself. And that particular idea is accepted widely and was accepted then. So in any case, we argued that sometimes there's a tension between trying to find answer a scientific hypothesis and making sure you address a clinically relevant question. So and we gave some examples, and in the conclusion to the article, we basically said that we believe that all clinical trials, at least clinical trials carried out by organizations like ourselves, had to be designed in a way that they could provide a meaningful answer to a therapeutic question, because that's what we were about. Our mission was to improve outcomes for cancer patients. But we might be able to answer biologic questions at the same time, but that should not subtract from doing what we were out to do, which was to find better ways of treatment, better forms of treatment. And we didn't think that all studies should have a explanatory objective imposed on them. So that was our working idea, and the approach we adopted, which I'll explain a bit more in the next slides, was that at the core of all our studies there should be a pragmatic component. There should be a relevant clinical question, and the trial should be designed in such a way as that we would get a reliable answer to that question, maybe negative, maybe positive. But we also were aware that there were all sorts of issues regarding the underlying biology of the disease in response to treatment that we might be able to shed some light on by adding to our core therapeutics trial the collection of other kinds of data that would help answer scientific hypotheses. So I had a chance to talk about this I guess our philosophy of how to do trials, and how our way of doing trials and thinking about trials met some important needs when I was asked to give the Cosby lecture in 1994. So we're still way back in time. At something that no longer at a meeting that no longer exists. There's no longer a meeting of the Royal College of Physicians and Surgeons where people present data, but there was back then. And the topic I took, and I can't remember whether I was asked to talk to it or decided to talk to myself, is that as always, it seemed there were challenges facing the development and conduct and successful conduct of clinical trials. And and we had to address those challenges. And these are taken from the slides I used at the time. I'm not going to go over these specifically, although the fourth bullet is important for what I'm going to talk about later, as is the fifth. And the other challenges were funding restraints from the healthcare system, concerns about quality, and competition. So what I did at that time is talked about a trial, and I'm going to use this trial as an example for a variety of things in this talk. A trial called BR10, some of you will be familiar with that study. And I called it at the time an Adapting to New Realities trial. And these are the characteristics of the trial that I thought suggested we were adapting. The trial addressed a therapy that was actually addressed in a meta-analysis, and it looked for meta-analyses that cisplatin-containing chemotherapy was beneficial in patients with non-small cell lung cancer, but the use of that treatment not really been carefully addressed in the setting that we were interested in, which is in patients who had their tumor removed and were potentially cured in what we call the adjuvant setting. And this is that's the clinical question we wanted to address. We had basis for wanting to address it, but no clear information about what the answer was going to be. The trial was multidisciplinary in structure. It was a surgically based study. So we had surgeons, medical oncologists, and importantly, pathology input in the person of Ming Chow. And we built into this study a strong correlative molecular biology component that included, in this particular case, snap frozen tissue, as well as paraffin embedded tumor banks. And there's a lot of interest in RAS back in that time, as there still is. And so we actually measured RAS at the outset of the trial and stratified on that basis. And we had lots of partners in the study, our own resources, and then we had to apply for grant funding for some of the other aspects from NCIC and from what was then MRC, now CIHR, and the company that made the drug we studied was also supportive. So this was a very straightforward study design. And I'm going to refer to this as a kind of standard type clinical trial, a simple parallel group comparison of an experimental regimen versus what was the standard of care. And at that particular time, the standard of care was observation only. So the study was a little bit more complicated in the sense that we required the acquisition of tissue. And as I already mentioned, we stratified on RAS status. But it was a simple one-to-one randomization between two forms of treatment at the core of the overall study. And the study wasn't all that easy to carry out. We started just at the same year that I talked at the Royal College. In 1994, it became clear as we moved along that we weren't going to be able to finish this study in Canada alone, although we had really strong support from the thoracic surgery community. And so we were able to get 3 US groups to join the trial, which meant it was a true intergroup trial. And we did enroll almost the required sample size over a 7-year period. Presented the results in April 2004 and published a paper in the New England Journal in June 2005. Those dates get a little bit relevant a little bit later on. So the results of the study were, to us, that we don't have many results quite like this. There was a substantial benefit of the use of adjuvant vinorelbine cisplatin in this setting. The difference in survival at 5 years, absolute difference in survival at 5 years was something like 13%. Later updated results was around 10%. So it was a major significant clinical outcome, and we're going to return to the significance of that later on in the talk. Just want to mention briefly that because, as I said, the core of the study was a clinical question, but we did collect tumor specimens, and that's had really an amazing output. in the form of something called LACE Bio. That immediately after the study was done, there were several studies of the same type being carried out internationally, and luckily the people involved in those studies decided to collaborate, both in terms of combining their clinical results, but also combining what they were able to learn from tumor tissue. And Leslie Seymour at our place has been a driving force behind this since it began. And this is just the title of a paper she wrote in 2019, which sort of summarized everything that was done in terms of immunohistochemistry on the paraffin-embedded tissues. But even further, the same group has now gotten an R01 grant from the NCI, and is now using genetic markers to look at the same tissues, and will also be able to validate the gene signature that was found in the frozen tissues or originally collected. So that work is ongoing and continues to be productive. So I think we can claim that we did both. We both answered a pragmatic question and also got some very interesting information on the underlying biology of the disease. So it seems like our formulation of how to go about doing both was reasonable. So during the time, though, that all these dates that I've shown you, the mid 1990s through the early part of the millennium, while we were doing that, the concept of what a pragmatic and explanatory trial also changed. And there hadn't been really, although we wrote that little letter a long time ago, there wasn't much of a literature about the distinction between pragmatic and explanatory. I go to meetings and you didn't hear people say, well, this is pragmatic, this is explanatory. The concept didn't seem to be all that important to people, but something changed in the mid 1990s, and there was an explosion of both articles and trials. Well, articles which mentioned pragmatic, that they were pragmatic, and trials that said they were pragmatic. Or this article was written just a little bit after 2010. So something changed, and there was all of a sudden a substantial interest in this distinction between pragmatic and explanatory. And I'm not 100% sure what changed or how it changed. There's in London, England, a research unit called the Pragmatic Clinical Trials Unit. And the director of that unit very conveniently has some videos on their website, which describes her perspective on the evolution of the concept of pragmatic and explanatory trials. And she cites this really short paper in the BMJ 1998 as being key to resurgence of interest. And what this paper did was conceptualize the distinction between explanatory and pragmatic trials a little bit differently than certainly we had held in our heads over the years before this. And what they did was basically equate explanatory trials with efficacy trials, and efficacy defined as the benefit of treatment under ideal conditions. And pragmatic trials were equated with effectiveness, the benefit of treatment produces in routine clinical practice. And so and we'll go on to see the use of both sets of terms, efficacy, effectiveness, and explanatory pragmatic, and at least for a time, both meaning each being equated with the other. I got kind of interested in this because I had always thought that the efficacy effectiveness difference was due to Archie Cochrane, who's the Cochrane collaboration is named after, and wrote a famous report back in 1971 talking about effectiveness and efficiency, random reflections on health services. But in fact, he doesn't use the term at all. He only uses the term effectiveness, which he refers to as what we learned from clinical trials and efficiency, which is what we learn from what happens when the results of those trials are applied in the health services. And the efficacy seems to have been developed as really more of a management term, going back to Peter Drucker, et cetera. But it's come to have a very clear-cut efficacy, especially a very clear-cut meaning in the medical context, meaning what it is here. That it's phrased differently sometimes, but basically, what happens when you design the conditions of your experiment, in this case, your clinical experiment, so that you maximize the chance of seeing a benefit from the intervention, as opposed to what would happen if you use that intervention in a more conventional circumstance. So I don't know, because I haven't really studied this, what which was the cart and which was the horse in this. But for sure, there became a substantial interest in this distinction. And some of the leaders of this were these two authors, plus a whole bunch of statisticians and trialists, many of them from Canada, like David Sackett and others, who got very interested in this distinction between pragmatic and explanatory. And this is a statement from an article from somewhere around 2008 that basically says that if we want evidence from trials to be used in practice, then the trials should make every effort to make their trial widely applicable, and which means that more trials should be pragmatic in attitude. And I just one comment is we're going to use the word applicable for most of the rest of this talk, because I really like that better than generalizable. Generalizable kind of has some of quasi-statistical meaning, whereas applicable is what we're really after. We want our trials to actually be useful. So anyway, they made that distinction, and they and many others then began thinking about, well, what characteristics of trials make them more explanatory? What characteristics make them more pragmatic? And they came out with an instrument called PRACY-1, and then refined that into something called PRACY-2. This is a picture of PRACY-2, which looks at 1, 2, 3, 4, 5, 6, 7, 8, 9 different characteristics of a trial. We're going to go through them a little bit toward the end of the talk, and you rate each of those, and then you connect the dots on each of those, and you can, if things are way toward the center, you're dealing with an explanatory trial. If things are way toward the outside, you're dealing with a pragmatic trial. And they were very clear that they didn't think this was a dichotomous distinction. There's a spectrum of trials which have both pragmatic and explanatory elements to them. And this is their verbal description of these characteristics. And I'll come back to this a little bit later on, so I let it sit up there a little bit longer. But this puts in words what's on that wheel. So in this kind of crude diagram, I think where things stood in sort of the mid 2010s was that people saw the trial world as consisting of trials that were either explanatory or efficacy, and trials that were either pragmatic and effectiveness in nature, that was spectrum. And that when we went to apply the results of trials in the real world, we could measure their effect either using Archie Cochran's term, which was efficiency, or more recently now talk about real world evidence. But the idea was you did the trials in the trial world, and then you applied the results in the real world. And underlying it was the idea that the more pragmatic the trial was, the more likely it was that you'd find real world evidence of benefit. Two things about this, so you can, at the bottom of this slide. Interestingly, Schwartz, Flamant, and Lelouch said explanatory trials were more generalizable than pragmatic trials, quite contrary to this, which I think is really because they had a slightly different idea in mind. I mean, their idea was that those explanatory trials tested an underlying biologic hypothesis, and if you had a positive result from that test, the biology might be the same in other circumstances. Whereas if you did a truly pragmatic trial, there was also that trial only applied to the population of patients who were included in the trial and to the interventions that were part of it, so that it only applies to one set of factors. And the other thing is that a lot of people who write about explanatory efficacy seem to imply that one can do a full-fledged, robust explanatory efficacy type of trial, and then move from that to replicating that in a phase 3 effectiveness trial. And although I think Sackett cites some examples from the cardiovascular literature, that particular progression in oncology is really very, very unusual. I'm not aware of any, or at least very many examples of a fully powered phase 3 efficacy trial was then replicated as an effectiveness trial. The usual progression in oncology, as you all know, is from phase 2 to phase 3, and that's what's still happening most often. So things didn't stop at that point. So that's where things were in 2015 or so. But this concept of what's a pragmatic trial has evolved further. And it's harder for me to find an article that talks about what the new idea of pragmatic trials are, because actually people don't seem to have consistent definitions. So I've just got some quotes for you. This is from an article in 2020 by a couple Spanish epidemiologists who basically say you can't measure effectiveness in controlled trials, that pragmatic trials are carried out in the real world and do not require randomization, because putting people on a study is a distortion, as they say, of usual practice. Now that's an extreme view, but it's one that's out there. There's something called the NIH Pragmatic Trials Collaboratory. And I just went to their website to see what they say, and they are focused on doing pragmatic clinical trials in the world of electronic health records, and hoping that we can answer questions in that setting, and then in that setting that would be truly embedded in practice, sort of like the registry type of trials that are happening out of Ottawa. A famous person, epidemiologist, whose name is well known in many contexts, has written about this too. And interestingly, he says that innovative clinical trials are explanatory by nature, and that truly the only pragmatic trials well, not the only, but the true pragmatic trials are comparative effectiveness studies, where two existing regimens are compared to each other. Where BR10 would fit on that spectrum, I'm not quite sure. And then there was a very good article in the New England Journal about pragmatic trials that points out, and it's echoing what was in the first quote, is that the conditions of practice that are different from the conditions of a trial, and particularly because people were approached and volunteered to be on a trial, so that it's often, to do a truly pragmatic trial, it's easier to do that in a setting where there isn't individual randomization and individual consent, which would be cluster randomized trials, where large unit groups like cities or hospitals are randomized rather than individuals. So sort of summarizing all this, there appears to be an emerging body of opinion that kind of sees pragmatic trials as over to the side somewhere, as a very special category of trials. And for them to really be as pragmatic as possible, they have to use somewhat different designs like cluster randomization, which I just mentioned, a step wedge design, which is a very clever idea where people are randomized to one treatment, but then are exposed to the other later on. Basically, an intervention is implemented in steps. And then large simple trials are often included as being more pragmatic than standard trials. All this is done to avoid deviation from standard practice. And usually the people who write about this say or imply that it's trials of this type, of this special kind of pragmatic type, that are the only ones that generate reliable evidence on the applicability of their results to clinical decision making. So this is my conceptualization of the world now. There's explanatory efficacy, but pragmatic effectiveness is somewhere in the middle between, it's still part of the trial world, is still part of the real world. And at least in some people's minds, that all trials are basically explanatory. And in order to find out whether the results of a trial really matter, you have to confirm them with real world evidence. So this seems to me to be a bit of a ways from the original ideas of Schwartz, Flamont, and Lelouch. And it raises the question of what evidence do we have concerning the applicability of trial results. Do we really know what kinds of trials are, how do we know, how much do we know about how trial results translate into practice? And of course, and that's otherwise known as the efficacy to effectiveness gap. And people have written about this. This is from Chris Booth and Ian Hannick, that in this particular article, they cite the evidence that it's often the case that patients treated in the real world with a drug that was found to be superior in a clinical trial don't do as well as the people who are on the trial itself. And I'll show you some more data like that in a second. And they attribute this mainly to these two things, is patient selection for trials, and that the fact that trials are done in a setting that's different from the real world. And we'll return to that. Another group of authors, another set of Canadians, looked at this same question and assembled some very, very interesting data in a very neat way, which is shown here. They had access to the new drug funding database in Ontario, where individuals who are provided funded access to when an individual is provided funded access to a new drug, their records are kept, and they can be linked to the outcomes for those individuals after they've been treated. So they looked at a wide number of new drugs, looked at the data from the new drug funding database, were able to calculate the survival of individuals on the new drugs in the real world, and then compared that to the survival of the patients on the trials that first established the efficacy or effectiveness of the drug. And what they found was that almost universally, people in the real world treated with a drug that was effective in a trial didn't live as long as the people on the trial themselves. And that's what they calculated hazard ratios, and that's what those things are to the right of the line. They point out, though, that the survival in the real world of a given innovative therapy is not as good as it was on the trial. But what a trial identifies what the trial identifies is the relative benefit of a therapy, not its absolute benefit, because the patients in the trial are not a random sample of the population. It's not like a survey. So what most trialists and most statisticians who write about trials say is what you can extrapolate or generalize or apply from a clinical trial is not the absolute effect you saw in the individuals on the innovative arm. It's the benefit of that versus the then current standard therapy, sort of the counterfactual of how much better does a person do treated this way than they would have done if they were treated with the previous standard therapy. And they point out that most studies that have looked at this have found that the relative survival benefit is preserved, but that's not an easy kind of research to do, which brings us back to BR10 because we have some information on that. After BR10 was completed, as I mentioned, the groups that had done similar trials collaborated and put their data together in a meta-analysis, which was called the LACE meta-analysis. And these are the studies. You can see there are 5 studies listed here BR10 is down at the bottom. And they all showed a benefit, but BR10 and Anita, which were the ones that used vinorelbine cisplatin and were kind of more conventionally designed clinical trials, showed a larger benefit. But there was no statistical heterogeneity in these data, and most people summarize the result of this meta-analysis as showing an absolute benefit of about 5% at 10 years, much less than BR10 showed. So the interesting thing is that Chris Booth and others did something that is not easy to do, which is to look at the impact of the adoption of adjuvant chemotherapy on a population level in Ontario. And they were able to show that in the years after that adoption, which was 2004 and on, there was a substantial improvement in the survival of patients whose tumors had been resected initially. And they comment that in fact, the observed absolute improvement, given that they had data on how many patients actually were treated, was more consistent with the results of Anita and JBR10. The J came in when the study became joint with the US, that they were more consistent with the results with JBR10 than the other two trials. So those real world findings on BR10, illustrate how powerful a role the powerful role real world research can play, say that 5 times fast, in assessing applicability of trial results. And as I said earlier, what we really want to know is the comparative benefit of an intervention. We want to know whether, if you're faced with a decision to treat a patient, you want to know if they'll be better off being treated with a new treatment as opposed to how it would otherwise be treated. It also, their work also illustrates how hard real world confirmation could be, because you have to wait until the intervention is implemented and then wait long enough till the outcomes for the patients treated with it have occurred. And so it was some time after the publication of the BR10 results that this work was able to be done. So calling for immediate real world effectiveness verification of trials work is a little hard. And as I mentioned before, it was what I called a standardly designed BR10 and Anita that were replicated in the real world data, not the large simple trials that were done at the same time and were a little bit more loosely designed, you might say. So this means to me that we need to think again, or hard, or continue to think about exactly what the characteristics of clinical trials done in the cancer world, and then particularly in the Canadian cancer world, are, and just how pragmatic can we make cancer trials, and can we make our existing standard kind of trials fairly pragmatic. So in closing, I'm just going to look quickly at these as the dimensions, the 9 dimension I mentioned before. The arrows point to the ones I'm going to talk a little bit more about, and for each of these, try to make the argument that what we're doing is pretty much the thing we should be doing. So taking these in turn, eligibility is a big issue, and there's not time in this article, this talk, to talk about it. There's no question that many clinical trials enroll a relatively select group of individuals, and probably a more select group of individuals than the trialists would want to see the results applied to, and we need to fix that, and people are trying to fix it. But I think you have to remember, and to some extent, I think the data that I showed you from Kelvin Chan supports, that it's always the case that trial participants are not going to be a random subset of a group of patients that the results are going to be applied to. Volunteers are always different. We know from voluminous literature in the late around the turn of the millennium, that there are lots of data showing that trial patients did better. The trial patients in an institution did better than patients at the same institution treated the same way. And for a long time, people thought there was some kind of magical trial effect, but I'm sure it's just a selection effect. So trial participants will never give us a full estimate of the benefit, the exact estimate of the benefit of population. And it's also the case that some of the criteria that have been used that limit trial enrollment really don't limit application. For example, the exclusion of people with previous malignancies doesn't really, from a trial, doesn't mean you wouldn't, if you had a patient with a cured carcinoma of the cervix, you wouldn't apply the trial results to their lung cancer. But that's a difficult and long conversation. I think the thing that we sometimes forget, that at least in Canada, most of our cancer trials are actually done in the settings where the results will be applied. They're actually done by the people who will end up applying the results, both applying them in their choices of therapy and sitting on committees that decide which therapies are going to be funded. So for cancer clinical trials, particularly radiotherapy and systemic therapy types of trials, the trials are being done where the results are being applied. That would be different from hypertensive interventions and things like that. But I don't think that should be forgotten. And similarly, although some trials require additional resources to be carried out, most of the trials are carried out using the facilities and personnel of the centers where the results will be applied. So I think we're closer to reality when we're doing trials in cancer trials in Canada. Oops, I carried over an arrow. So the people argue that flexibility in treatment administration is a virtue and makes a trial more pragmatic because doctors make variable decisions in applying treatment in practice. But I'm not quite so sure it's a virtue in the cancer treatment setting, where it's probably more useful to have the results of a trial which clearly describes how the treatments were given and what adjustments were made for toxicity, et cetera. So may not be a virtue in a cancer trial. Primary outcomes is a big issue, obviously, and the whole issue of surrogate endpoints, et cetera. But I think we can design trials that do address a meaningful endpoint and of sufficient magnitude to be meaningful, and that's been studied a bit. And almost all cancer trials use an intent to treat analysis. So on many of these spokes on that wheel, conventional cancer clinical trials meet the definition, or at least show signs of pragmatism. So to conclude, there is a widespread interest in pragmatic trials, but I think if you looked at the quotes I said, you a ton of different ideas about exactly what constitutes a pragmatic trial and what they're supposed to do. And it does seem that this was a point made in one of the articles, that people just want to put the tag pragmatic on a trial they're doing for a variety of reasons, some of which don't hold up. But the issue really, to me, isn't nomenclature. It doesn't matter what we call things. What we really care about is whether the results of trials actually will be applicable in the setting where they're aimed. And I would argue that in the Canadian cancer setting, at least, inventing a whole new category of trials of different design is not necessarily the best way to get applicable results. As certainly as you can see from the BR10 example, conventionally designed clinical trial has clearly applicable and important results. So I think, and this is, of course, kind of the be conservative of me, is that we should actually go back to what Schwartz, Clement, and Luce were actually talking about, and make sure that the typical design trials are as pragmatic, again, in their original sense of the term, as we can. And thank you very much for your attention. That is it.
Thank you very much, Dr. Patter. That was fantastic. Perhaps I can open up the floor at this point for some questions.